All right, welcome back to the deep dive. Today we're tackling something truly foundational, something you can't escape if you're into biology at all.
Going deep, are we?
Deep dive, you could say, the central dogma of molecular biology, no less.
The classic DNA to RNA to protein pathway. Every bio student's first love.
Exactly. But we're not just going to regurgitate the textbook here. We've got this incredible material that really unpacks how this flow of information actually works, the molecular gears turning.
The nuts and bolts of life itself.
Love it. So, mission for this deep dive is how does our genetic blueprint, that DNA, actually get used to build all the stuff, all the machinery that makes a cell run?
Ambitious, I like it.
Well, gotta start somewhere, right? And that starting point's gotta be DNA itself, the big boss molecule.
No arguments there.
Yeah?
DNA, that elegant double helix, that's where the story begins. Two long, intertwined strands, each a sequence of those chemical units, the bases.
And they're like mirror images, aren't they?
Complementary, yes. The sequence of one strand totally dictates the other. And a crucial detail. They run in opposite directions, anti-parallel.
Anti-parallel. Always gotta picture that in my head to remember it.
But that arrangement's key. It impacts how this information's read, how it's copied, the whole shebang.
Okay, but seriously, two long strands just chilling, entwined. Seems like they'd fall apart if you looked at them wrong.
Well, there's some serious chemistry holding them together. Hydrogen bonds between specific pairs of those bases. Adenine, always with thymine,
Hanging two.
and guanine with cytosine, G and C. Now, one hydrogen bond, puny. But think about it, millions of these along the entire DNA molecule, suddenly you've got some real stability.
So strength in numbers.
Precisely. And little side note, in RNA, which we'll hit later, it's uracil, U, instead of thymine.
Gotcha, mental note made. So it's the order of these bases, like A, T, G, C, whatever, that actually spells out the instructions, right? Like a code.
That's it. The sequence is the language of life. Tells the cell what to build, how to build it, when to do it. And holding all this together, literally, is the backbone of the strand. Sugar and phosphate groups, alternating, linked by those strong phosphodiester bonds.
Sturdy scaffold. But here's what always gets me. DNA, it's got to be huge GE, right? All that information. Yet it fits inside these microscopic cells. How is it even possible?
Supercoiling, my friend.
Yeah.
Think garden hose stuffed into a tiny bucket. You got to coil it tight.
Hmm.
Enzymes called topoisomerases, they're the ones doing the coiling.
Yeah.
In Bacteria and Archaea, it's DNA gyrase that really cranks it up. Negative supercoils packs that DNA in tight.
Genius. So we've got this super organized DNA in our chromosome, but the story doesn't end there, does it? There's more to genetic info than just that.
You're sharp. Beyond the chromosome, you've got plasmids, little circles of DNA replicating on their own, separate from the main show.
Renegade DNA, huh?
Kind of. Often they carry genes that give this cell a leg up. Antibiotic resistance, for example. Those are called R plasmids. And they're a big reason why antibiotic resistance is such a problem these days.
Yeah, heavy stuff. And of course, we can't forget viruses. They've got genetic info too, but a bit different.
Viruses are like little genetic couriers, DNA or RNA, packaged in a protein coat, just hopping between cells.
Sneakily spreading their information.
Exactly. They hijack the cell's machinery, make more copies of themselves, a whole other way to transfer and spread genetic info.
And lastly, those mysterious jumping genes. Transposable elements always sounded kind of sci-fi to me.
Yeah. These guys are DNA segments that,
Yeah.
they jump around the genome, insert themselves into different spots.
Like genetic nomad.
Perfect analogy. And their jumping can change things. Alter gene expression, even disrupt a gene entirely. Adds a whole layer of complexity to how the genome evolves over time.
Wild. So we've got the DNA blueprint in all its forms, but how's it actually used? Gotta make copies first, right? That's DNA replication.
Right on. Before a cell divides, got to make sure each daughter cell gets a full set of instructions. And replication, it's semi-conservative.
Semi, meaning?
Each new DNA molecule, it's not brand new. One strand's from the original, one's newly made. Like keeping the master blueprint while making a copy.
Ah, clever. And this copying, it just starts anywhere, or?
Nope. Specific spots on the DNA, origins of replication, ORIS for short. The molecular starting line.
Found the starting line. Now what? Just unzip and copy.
First, got to unwind that helix. DNA helicase, that's our enzyme for the job. Like a zipper, breaks those hydrogen bonds between the bases, separates the strands.
Unzipping the DNA.
And of course, those strands want to snap back together. That's where single-strand binding proteins come in. They hold the strands apart, keep them stable, ready to be copied.
Templates prepped. Now for the main event, the copying itself. DNA polymerase, it's a star, right?
The star indeed. DNA polymerase adds those DNA building blocks, the nucleotides, to the growing new strand. But here's a catch. It can only build in one direction, 5 to 3 foot.
5 to 3 foot, 5 foot to 3 foot. Got to keep reminding myself. Why is that a problem?
Because those DNA strands, remember, anti-parallel, running opposite ways. So one new strand, the leading strand, it's smooth sailing, builds continuously as the DNA unzips.
Easy peasy.
The other strand, the lagging strand, it's a different story. DNA polymerase can't just run backwards, it has to build in segments.
Like paving a road in reverse, got to do it in chunks.
Perfect. Those segments, those are our Okazaki fragments, short stretches of new DNA on the lagging strand.
Okazaki fragments, those are famous.
And even starting those fragments, DNA polymerase needs a little help, needs an RNA primer laid down by the enzyme primus, gives it that 3-footer end to start building from.
So we've got these chunks of DNA with RNA primers in between. How do we end up with one nice, complete strand?
That's DNA ligase to the rescue. Once those Okazaki fragments are made, the RNA primers get replaced with DNA. And ligase, it's like molecular glue, joins those fragments together, makes one continuous strand.
Problem solved. Elegant, really. Now, in bacteria and archaea, their DNA is circular, right? How does replication work on a loop?
They start at one origin, and replication goes both ways. 2 forks moving around the circle, like a bubble expanding.
Replication bubble. I like it.
Until those forks meet up at a specific spot, the terminus. Then, bam, 2 new circular DNAs ready to go.
Lots of moving parts here. Is there a name for this whole replication crew?
Replicum, the whole shebang, all those enzymes working together at the fork. And of course, got to make sure those copies are accurate. DNA polymerase has this amazing proofreading ability.
Checks its own work.
Exactly. It can backtrack, remove any wrong bases it added, keeps those errors super low.
Quality control is key. DNA's been duplicated, great. Now, how do we go from info to actual stuff, the proteins? That's transcription, right?
That's the next step. Transcription is making RNA from DNA, like taking notes from the master blueprint.
Yeah.
And RNA polymerase, that's our enzyme for this job.
Another polymerase?
This one doesn't need a primer, though. It finds these specific DNA sequences, promoters, that say, hey, gene starts here.
The start here signs for RNA polymerase. But how does it even find those signs in bacteria?
They've got this helper, the sigma factor. It guides RNA polymerase to make sure it binds to the right spot on the promoter.
Like a parking attendant for enzymes?
I like that. And bacteria, they have different sigma factors, each recognizing different promoters, so they can control which genes are transcribed depending on what's going on around them.
Clever adaptation. So RNA polymerase is chugging along, making RNA. How's it know when to stop?
It hits these DNA sequences called terminators, like a stop sign. RNA polymerase, let's go, releases the new RNA, and that's it for that gene.
Makes sense. And unlike replication, where the whole genome gets copied, transcription's pickier, right?
Very much so. Usually just specific genes or groups of related genes are transcribed. Only what's needed at that time.
Efficient. So, new RNA molecule hot off the press. What kinds are there, and what do they do? Specifically for protein synthesis, I mean.
The big 3 are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Messenger RNA, that's the one carrying the DNA's message, the protein blueprint, to the ribosomes.
Ribosomes, those are the protein factories.
Exactly. Now, in bacteria and archaea, one mRNA can sometimes code for several proteins. Polycistronic mRNA, it's called. Comes from groups of genes called operons. Super efficient way to coordinate making proteins that work together.
Multitasking RNA. What about tRNA and rRNA?
Transfer RNA, those are the little adapters. One end recognizes a codon on the mRNA, 3 bases that code for a specific amino acid. The other end of the tRNA carries that amino acid.
So they match the code to the building block.
Precisely. Link between the mRNA instructions and the protein's sequence.
Mm-hmm.
And ribosomal RNA, that's part of the ribosome structure itself, helps with the whole protein synthesis process.
We've been talking bacteria, archaea. What about us eukaryotes? Are transcription any different?
More complex, for sure. Happens inside the nucleus, and there's this whole RNA processing step beforehand. Before the mRNA leaves the nucleus, it gets a cap on one end, a tail on the other, and any non-coding bits, introns, get cut out.
RNA getting a makeover.
Right. Interestingly, archaea's transcription kind of resembles eukaryotes more than bacteria, even have introns sometimes. Shows how evolution works in mysterious ways.
So mRNA is made, processed if needed. Finally, the moment of truth, translation, RNA to protein.
For the grand finale, translation happens on ribosomes, those RNA protein complexes. Bacteria and archaea have 70S ribosomes, eukaryotes have 80S, a bit bigger.
S for Svedberg unit, right? Measures how fast they sediment.
You got it. Ribosomes have two subunits. Small one grabs the mRNA, large one does the actual protein building.
And how's it know which amino acid to add and in what order? Got to be following the mRNA, right?
Absolutely. Ribosome reads the mRNA in codons, 3 bases at a time. Each codon specifies an amino acid. That's where our tRNAs shine. Their anticodon matches the mRNA codon, and they bring the corresponding amino acid.
So tRNA is like the delivery service, bringing the right package based on the address.
Perfect. But how do they get the right package in the first place? That's those aminoacyl tRNA synthetases. Each one's specific for amino acid and its tRNA. Make sure the right amino acid gets loaded onto the right tRNA.
Quality control again. Now, 4 bases, 3 at a time. That's 64 possible codons, each one unique.
Not quite. The genetic code's degenerate, meaning some amino acids have multiple codons. 61 codons for amino acids. 1 AUG is the start codon, also codes for methionine. And then there's 3 stop codons, UAA, UAG, UGA. They signal end of protein.
So it's like punctuation in the genetic sentence.
Exactly. And the ribosome has to read it in the right frame, start at the right AUG, read 3 at a time. If it gets shifted, the whole protein's messed up.
Catastrophic frame shift. And what about this wobble I keep hearing about?
That's about the third base in the codon, a bit more flexible pairing with the tRNA, because one tRNA can recognize a couple different codons. Less strict, more efficient.
Got it. mRNA, ribosomes, charged tRNAs. Lay it on me. How's translation actually happen?
3 steps.
Yeah.
Initiation, elongation, termination. Initiation, small ribosomal subunit grabs the mRNA. In bacteria, they've got this ribosome binding site that helps it find the right spot. Initiator tRNA, carrying methionine, binds to the start codon. Then the big subunit joins, party started.
Ready to build.
Elongation, that's where the chain grows. tRNA matching the next codon comes in, binds to the A site. Peptide bond forms between its amino acid and the growing chain. Then ribosome scoots down the mRNA, translocation. tRNA with the chains now on the P site, old tRNA exits. Rinse and repeat.
Like an assembly line, adding parts one by one.
And finally, termination. A stop codon shows up, no tRNA recognizes it. Instead, release factors bind, signal, cut the chain loose, protein's released, ribosome splits up.
It's mind-blowing, these tiny machines building all the proteins. And multiple ribosomes can work on one mRNA at the same time, right?
You know what? Polysomes, they're called. Makes protein production super efficient. And fun fact, RNA is not just structural, it helps catalyze those peptide bonds too. Real team player.
RNA doing it all. There's even that tmRNA, right? For when things go wrong.
Ah, the rescuer. tmRNA helps ribosomes that get stuck on messed up mRNAs, ones without a proper stop codon. Saves them from getting jammed.
Whew, good save. So protein's made, but it's not necessarily ready for action yet, right? Gotta process it, fold it, send it to the right place.
You got it. Lots of proteins need post-translational modifications. Folding, with chaperone proteins helping out, adding cofactors, other tweaks to specific amino acids.
Like fine-tuning the protein.
Exactly.
Yeah.
And then got to get them to the right spot. Some stay in the cytoplasm, some go to membranes, some get secreted out of the cell entirely.
How's the cell know where each one goes, like a protein GPS?
Many proteins have signal sequences, little amino acid tags, usually at the beginning. Like a shipping label tells the cell, send this one to the membrane, or this one goes outside.
Makes sense. So how do proteins actually cross the cell membrane in bacteria and archaea?
2 main systems, sec and tat. Secs for unfolded proteins. Often chaperones keep them unfolded till they reach the membrane channel. Tats for pre-folded proteins, uses more energy.
Different pathways for different needs. Now, gram-negative bacteria with their extra outer membrane, that's got to complicate things.
It does. They've evolved these sophisticated secretion systems, 6 main types, type I to 6, get those proteins across the outer membrane, sometimes even inject them into other cells.
Whoa, protein weaponry.
Right. Some are 2-step systems, like type 2 and V, use sec or tat to get to the periplasm, then another mechanism for the outer membrane. Like type V autotransporters, they have a built-in part that helps them cross.
Self-sufficient proteins.
Then there's the 1-step systems, types I, 3, 4, 6. They span both membranes, direct delivery.
Those injectosomes we talked about, which type are they?
Those are type 3. Syringe-like? Inject proteins right into host cells. Many pathogens use them to mess with the host processes, helps them survive and cause disease.
Sneaky. Well, what about the other one-step systems?
Type 4, very versatile, can move proteins, even DNA, involved in conjugation, bacteria swapping DNA. And type VI, they're like little harpoons, shoot proteins into other bacteria or eukaryotic cells.
It's like a whole microscopic battlefield out there.
It is. And these secretion systems, they're key for bacteria, survival, getting nutrients, causing trouble. Understanding them is huge for fighting bacterial infections.
Wow, this deep dive has been incredible. From DNA structure to protein delivery systems, we've covered it all. The whole flow of genetic information, just mind-blowing.
Our goal was to summarize the whole chapter, all the key points, everything about molecular information flow and protein processing. DNA replication, transcription, translation, protein targeting, even those complex secretion systems. We really went through every bit.
It's humbling, really. All these tiny processes working flawlessly inside every cell. And if anything goes wrong, well, that's often when disease happens. But understanding these processes, that's how we find new ways to treat diseases, develop new therapies.
Truly. The elegance and power of life at the molecular level. Amazing, isn't it?
Absolutely. Thanks for joining us on the deep dive. And until next time, keep those minds curious.